last few classes we are dealing with the, what are we dealing with? The central nervous system. Okay. So what are the topics we have covered here? What are the topics covered in the central nervous system? अभी तो क्लास शुरू हुई 4 दिन हुआ है, किताब पलटने की बात है क्या? What are the topics covered? Organization of central nervous system? Yes? Yes or no? अभी एग्जाम में CNS नहीं है इसलिए किताब नहीं खोली जा रही है, जब फाइनल में आएगा तब तक भूल जाएंगे. So we have covered with the organization of central nervous system, the receptors, reflexes and synapse. So what do you mean by a synapse? What does it mean by a synapse? We have already learned this at some other place also. Synapse. Yes, answer. Okay, so if she is talking about the neuronal synapse. Synapse can be at the neuromuscular junction. Have you recollect this? Do you know that? We have studied about the neuromuscular junction. So that was which type of synapse? A chemical mediated, because there was the release of the chemical acetylcholine. So this is one of the chemical types of synapse. Now the other type of synapse is electrical type of synapse, and this takes place through the gap junction. Now where did we study gap junction? That is the intercellular communication. We studied at desmosomes, hemidesmosomes, tight junctions and gap junctions. So give me an example of a gap junction. Answer. I don't want to raise hands. Do you know the answer? Intercalated disc, which is found in the cardiac muscle. The second example of gap junction. Second example. Which muscle? Apart from the cardiac muscle? Single unit smooth muscle. So this is a synapse, or synapse is an area where there is transmission of some activity for further propagation. This we have dealt and sir has taught you this. So we move on to the various types of synapse. So this was the mechanism of synaptic transmission. This was a neuron. This can be an axon. This can be a muscle, it can be anything depending on the junction which we are dealing with. But right now, since we are dealing in the CNS, it is a axon or a dendrite. It can be an axon versus an axon. It can be an axon versus a cell. So these are the various types of classification in the central nervous system for the synapse. So we have the synaptic cleft, we have the diffusion of the neurotransmitter. Now what are the neurotransmitters in a central nervous system? Do you think it will be acetylcholine? Do you think it will be acetylcholine? Now what is the purpose of this synapse in a central nervous system? What is the function of synapse in a central nervous system? वो क्यों है? कुछ तो रोल होगा उसका. क्या करेगा? वो इम्पल्स का जनरेट करेगा. अगर आपको टच किया आपने गरम चीज को इन prick, so what is the response? You withdraw your finger, that's very common. Now what was the reaction? Was the impulse sent to the muscles that you have to go for contraction. So what was the impulse generated? Whether it was an excitatory or whether it was a inhibitory. What was it? Excitatory for which side? This is, you have learned the flexor withdrawal symptoms. फ्लेक्सर विड्रॉल सिम्टम पढ़ा है ना आपने, फ्लेक्सर विड्रॉल रिफ्लेक्स. आप चलते चलते आपके पैर में कांटा चुभा तो आप क्या करते हो? यू बैलेंस इट. सो व्हाट आर यू डूइंग? दिस इज गोइंग इनटू फ्लेक्शन एंड द अदर साइड इज गोइंग इनटू extension. So what is it? One side there was a excitatory impulse and the other side it was a inhibitory impulse. Yes? So in that manner the whole purpose of this synapse taking place in the central nervous system is either it is excitatory in function or it is inhibitory in function. So this is just a recap of what sir has told you. The postsynaptic membrane, it binds with the receptor. So receptor can be ligand gated, voltage gated, or what? Protein link gated. Okay. So if it is ligand gated, so it means acetylcholine. If it is protein linked receptors, you have opening of the ionic channels, you have sodium coming in, depolarization occurring, and you have a excitatory impulse. But suppose if it goes into the favor of potassium getting in and chloride getting in the neuron, what will happen? क्या होता है? अगर हम एक्शन पोटेंशियल को याद करें तो डीपोलराइजेशन क्यों होता है? सोडियम बहुत सारा इन्फ्लक्स हुआ, तो इसी तरीके से यहाँ पे भी सोडियम इन्फ्लक्स होगा तो क्या होगा? एक्साइटेटरी. सो यू हैव एक्साइटेटरी पोटेंशियल, दैट इज पोस्टसिनैप्टिक EPSP, excitatory postsynaptic potential. And what is IPSP? Inhibitory postsynaptic potential. So if I want, or if the impulse is there to potentiate an excitation, what will happen? Sodium channels open. And what is released? Excitatory neurotransmitter. What is that? CNS. What is it? Okay, that we will come to it later on because neurotransmitters abhi hua nahi hai. So have you understood the synaptic transmission? Postsynaptic membrane पे गया, सोडियम चैनल्स ओपन हुए या आयोनिक चैनल्स ओपन हुए, साइक्लिक AMP फॉर्म हुआ, इंट्रासेल्युलर एंजाइम ट्रांसक्रिप्शन, एंड देन यू हैव द एक्शन पोटेंशियल being generated. Whenever there is depolarization, it is EPSP due to the presence of sodium channels. IPSP, this is hyperpolarization, that is efflux of the potassium from the cell to from the neuron here. Now we are not talking about a cell, we will be talking in the terms of neuron. So from the neuron it will go to the ECS, and also chloride, they will move. So you will have the hyperpolarization curve. If you recollect the curve, it is usually like this. When it goes beyond the resting membrane potential, it is a hyperpolarization and closure of the sodium channel. Now local responses, this we have seen. These are the properties of synapse which you have already studied. It is a one-way conduction. It does not follow all or none response. Slowly, even if you give the impulse, it will generate and it will come into a excitation or inhibition, whatever is there. So it's a graded response. It can be summated. All or none law means either it's a yes or it's a no. Then you have, it can be either depolarizing or it can be a hyperpolarizing type. Now what are the, depending on the properties of the nerve fibers also, you have the impulse. It can be length constant, it can be time constant. Temporal summation, time constant. टेम्पररी है, तो हमने 10 सेकेंड तक उसको इम्पल्स दिया, जितना हुआ समेट होके इट गिव यू एन एक्शन. आफ्टर 10 सेकेंड वी डिड नॉट गिव एनी इम्पल्स. स्पाशियल लेंथ है, स्पाशियल मीन्स आराम से हम उसको दिए जा रहे हैं इम्पल्स, बिकॉज आवर कंसेप्ट इज कि यहाँ से इम्पल्स यहाँ तक पहुँचना है, तो समय की पाबंदी नहीं है, तो हमें ये लेंथ कवर करना है. और जब टेम्पोरल समेशन होता है, समय की पाबंदी है, तो हमें उसी समय हमें ज्यादा से ज्यादा इम्पल्स करना, ठीक है। सो दिस इज अबाउट द टेम्पोरल समेशन एंड द स्पैशियल समेशन. सो सम अप ऑफ द प्रॉपर्टीज, वन वे कंडक्शन, सिनैप्टिक डिले, ये सब आपको रिवाइज हो रहा है, यस और नो? ये सब सिर के ऊपर से जा रहा है. सिनैप्टिक फटीग, व्हाट इज सिनैप्टिक फटीग? ये कहाँ पढ़ा आपने? विच प्रॉपर्टी? स्केलेटल मसल, विच प्रॉपर्टी? जेनेसिस ऑफ फटीग. यू वेंट ऑन गिविंग इम्पल्स. अगर हम आपको बम्बार्ड करते जाए CNS टॉपिक से तो क्या होगा आफ्टर हाफ एन आवर? दैट इज एग्जॉस्शन, right? And it happens with you all. By the time you reach home at 5 o'clock, what happens? You are totally exhausted. That is the word you use. And then what do you do to bring back that exhaustion? Either you lie down, you switch on the TV, your favorite channel, because वो मोबाइल पे थोड़ा थोड़ा आग गड़ा के देखना पड़ता है, लेकिन वो दिखता है, दिन भर भी देख सकते हो. But then it happens, you try to remove and you try to restore your capacity. So that is restoration, that is apart from this, but just to explain what is exhaustion. Then electrical phenomena, this I have discussed. EPSP, excitatory postsynaptic potential, depending on the sodium channel and decreased conductance of potassium and chloride. IPSP, that is inhibitory postsynaptic potential. It is more because of the efflux of the potassium channels, chloride channels, and closure of the sodium gates. So we have EPSP, summation, effect of pH, all these matter to the EPSP. If the atmosphere is conducive, so we will definitely have an excitatory conduction, if at all. If you want an impulse which is to be excitatory, but the pH is not working, if the receptors are not working, there is no collaboration. So what will happen? You will not have a proper EPSP in spite of you giving or you are trying to have a excitatory potential. So these factors matter a lot. Body response. So if you say कि हमको हमेशा ही excitatory response है, now what happens in Parkinson's? What happens? The patient has a twitching. So हमें वो twitching permanently थोड़ी चाहिए. We need the inhibition, but there is a decrease or there is a disorder in the pathway which is inhibiting this inhibitory process. So what are we having? The patient is just doing this way. So this excitation is not under your control. Get it? So when we talk about the factors responsible, so we have to have the entire atmosphere conducive, internal body atmosphere. Drugs, effects of hypoxia, drugs and post-tetanic potentiation. Now what is synaptic inhibition? अब एक synapse है. You have, what are the components of a synapse? What are the components of a synapse? I need 3 children. Come out. कोई भी आओ. I'll just try to portray it in the manner. आ जाओ. उठ के आओ. मेरे mentees आ जाओ. चलो, क्योंकि वो तो मना नहीं करेंगे. उठ के आओ. बाकी तो उठेंगे नहीं. एक और बच्चा आओ. चलो. One more. Come. तुम आ जाओ. Now भैया. Now routinely what is happening is, this is the presynaptic, this is synaptic cleft, and this is the postsynaptic. Right? अभी तुम हटो साइड से. यहीं रुको. Now you get an impulse. What happens? Calcium open, sodium open, neurotransmitter release, goes in the synaptic cleft. Synaptic cleft में postsynaptic membrane है. which enhances the sodium channels, potassium channels, whatever, and you get a response, right? Now when I talk about synaptic inhibition, what it means is synapse पे रोक लगाना, yes or no? ठीक, मुझे नहीं चाहिए ये सिनेप्स पे कोई आगे काम हो, तो वो रोक कहाँ कहाँ लग सकती है? एक हो गया पोस्टसिनेप्टिक और एक इधर। अब इधर कैसे लगेगा? एक्शन पोटेंशियल तो रिसेप्टर पे आ ही गया, तो इसमें होता है एक चैनल अटैच, एक और एक्सॉन आके इसको अटैच किया, तो दिस बिकम्स प्री सिनेप्टिक इनहिबिशन। ये जो इम्पल्स दे रहा था इसको नहीं देना है, तो जो इसका आधा इम्पल्स किसी और से आ रहा होगा वो एक्साइटेशन में है, कोई इश्यू नहीं, बट दिस विल स्टॉप गिविंग द इम्पल्स, सो दिस बिकम्स अ प्री सिनेप्टिक इनहिबिशन। क्योंकि इफ आई कंसिडर अ न्यूरॉन, जरूरी नहीं कि वो सिंगल चैनल न्यूरॉन आ रहा, वो मल्टीपल एक्सॉन्स को भी कनेक्ट कर सकता है, मल्टीपल सेल स्ट्रक्चर नर्व्स को कनेक्ट करके हम एक मोटी नर्व को ले रहे हैं, तो उस एक नर्व में अगर एक ने बोल दिया मुझे काम नहीं करना, तो दिस इज द प्री सिनेप्टिक इनहिबिशन व्हिच ही इज प्रोवाइडिंग। सो इसका इफेक्ट कम होगा कि ज्यादा होगा? कम होगा। तो ओवरऑल you may have a presynaptic inhibition. Now when I talk about the postsynaptic inhibition, what will happen? उधर से impulses आ रहे हैं, IPSP हो गया कि वो आए impulse negative, मतलब सारे तरफ से presynaptic पे जो impulses आए they were of the inhibitory variety and you had a IPSP being generated, that is efflux, तो वो direct हो गया, right? So this is how synaptic inhibition works, right? चलो, so I'll come back to the topic. So you have feedback inhibition. Now feedback inhibition कहाँ से आएगा? हमने तो अभी सिनेप्स की बात की लोकल एरिया पे, फीडबैक कहाँ से आएगा? अगर तुमसे फीडबैक लिया तो कहाँ से आएगा? मैंने क्लास लिया, तुमने क्लास अटेंड किया, अब तुम बाहर चले गए, फीडबैक क्या दोगे? अच्छी थी, बुरी थी, डांट पड़ी, नींद आई, मस्त रहा, एसी में बैठे, हाँ, ये फीडबैक है। तो ये फीडबैक इनहिबिशन आएगा कहाँ से? स्पाइनल कॉर्ड से। दैट इज व्हाट वी टॉक्ड अबाउट द रिफ्लेक्स। एक इम्पल्स हुआ, एक रिसेप्टर स्टिमुलेट हुआ, चाहे पेन रिसेप्टर स्टिमुलेट हुआ, कुछ हुआ, वो स्पाइनल कॉर्ड के लैमिने पे जाके अपना इम्पल्स दिया, इंटरन्यूरोनल स्ट्रक्चर बना, विच नर्व फाइबर वाज स्टिमुलेटेड? मसल स्पिंडल 2A, 2B. Okay. So what happened? You had the pinprick, गया, रिसेप्टर आया, रिफ्लेक्स आर कैसे बनता है? There is an effect, there is a synapse and there is a motor organ. तो सेंसरी गया, उधर से वापस आया, बाहर आ, तो फीडबैक किसने दिया? स्पाइनल कॉर्ड ने दिया कि बस चोट ठीक हो गई है, नो मोर रिक्वायर्ड, so the feedback inhibition. Feed forward inhibition is by the cerebellum. अभी हम cerebellum cover नहीं किए, but when we read cerebellum, you will remember that it helps in the feed forward inhibition, which maintains your equilibrium, posture, and whatever are the functions. Okay, and this helps. This takes place with the help of granular cells, Purkinje cells, basket cells. These are all the various types of cells found in the cortex of the cerebellum. Right? Now we come to the inhibition per se. Postsynaptic I spoke, direct and indirect. So what happens in a postsynaptic inhibition? Muscle is stretched. That is what we discussed. Either it goes into flexion or it goes into extension. So there is a motor neuron, muscle contraction, Golgi apparatus which cause the inhibitory response, and antagonist muscle is inhibited and the muscle in which the injury was there, it is relaxed. So when I am walking, if I get a stumble by a stone, what I do? I will just go like this. So what is happening? The entire pain is taken on the antagonist muscle, and the actual muscle, agonist muscle, goes into relaxation, right? So this is about the postsynaptic inhibition. Then when we have the feed forward, you have the Renshaw cell inhibition, the recurrent inhibition, and that leads to the indirect. ये तो डायरेक्ट हुआ कि चलते चलते चोट लगी, हम लटक गए, इधर से हो गए, ये स्ट्रेच हो गया, so that's a normal postsynaptic inhibition. So what happens in the indirect? Previous firing हो रही थी negative की, more of impulse went on, so hyperpolarization continued. So that is an indirect postsynaptic inhibition. We had already given the inhibitory impulse, interneuron से inhibitory impulses आए थे, तो हमको पता चला कि antagonist muscle को stimulate करेंगे, वहाँ पे उसका excitation होगा और यहाँ पे muscle relax होगी, clear? तो अगर ये firing continue होती रही, तो what will happen? Already inhibition है, so what will happen? It will be further continued. So it is an indirect type of postsynaptic inhibition. This is very complicated a little because you have to read this to understand. So please go home and read it, otherwise by the time for your PUC you will tend to forget it. So just make it a point that you will read it at home. Now this inhibition takes place, I have repeatedly been telling, because of the fluoride and the potassium channels. Now I talked about the presynaptic inhibition. What happens? There is an axon and there was a cell body or a dendrite or axonal synapse. So it occurs at the axonal synapse. The same procedure, presynaptic inhibition, that axon decides to have the liberation or the release of potassium and chloride ions. So what will it lead to? Inhibition at the presynaptic level, and thus leading to the total inhibition. The neurotransmitters which are involved here are GABA. GABA is what? Inhibitory neurotransmitters. So if I talk about this is an excitatory axon which was coming, and this was the dendrite on which the synapse was taking place. Now this was that fourth bacha who stood that he wanted to inhibit this excitatory neuron. So this presynaptic membrane, now this becomes the postsynaptic membrane for this synapse. How many synapses are there in this diagram? 2. This is synapse, this is postsynapse. But this is already an excitatory neuron which is a presynaptic membrane for this synapse. So this presynaptic will decrease the inhibition here, or will cause an inhibition, so the excitation of this axon decreases. So ये major synapse है जो हमें चाहिए रिस्पॉन्स, लेकिन इसके साथ एक एक्सॉन अटैच हो गया जिसने इनहिबिटरी इम्पल्स भेजे, तो फाइनली क्या होगा? जो प्री सिनेप्टिक इनहिबिशन आ रहा था या प्री सिनेप्टिक एक्शन पोटेंशियल आ रहा था, उस पे रख गई रोक. आप क्लास जा रहे हो, 2 बच्चे आ रहे हैं क्लास में, तीसरे ने बोला नहीं जाएंगे, तो एक एक कदम रुक गए. क्या करना है? जाके लाइब्रेरी चलते हैं, पेपर फ्राई चलते हैं. क्या होगा? प्री सिनेप्टिक इनहिबिशन. अच्छा खासा क्लासरूम में आ रहे थे, कुछ तो सुनते, लेकिन एक बंदे ने आके सारा वो कर दिया. पिकनिक पे जा रहे हो, 4 लोगों का ग्रुप बना, हैप्पीली वन डिसाइड्स कि मैं नहीं आऊंगा, पूरा प्रोग्राम चौपट. सो दिस इज प्री सिनेप्टिक इनहिबिशन, राइट? नहीं तो मम्मी ने रोक लगा दी, वो तो सबसे मोस्ट कॉमन कॉज ऑफ प्री सिनेप्टिक इनहिबिशन है. Yes or no? Yes. देखो, honestly. So have you understood the mechanism of presynaptic inhibition? Please don't cite these in your exams. So presynaptic inhibition में same process, opening of ion channels, hyperpolarization, decrease in the magnitude of action potential. Now since it's a presynaptic inhibition, calcium influx जो हो रहा था presynaptic में टर्मिनल में वो कम हो जाएगा, तो acetylcholine release भी कम हो जाएगा. If I talk about the neuromuscular junction, but if I talk about here, तो अगर उसमें neurotransmitters excitatory वाले रिलीज होने थे वो भी कम हो जाएंगे. So indirectly there will be a decrease in the excitation process. Allow a particular excitatory input to be inhibited without affecting other excitatory synapse. तो जब आपका ग्रुप बना, सपोज पिकनिक का, एक ने अड़चन डाल दी कि मैं नहीं जाऊंगा, आप बोलते हो ओके फाइन, rest all of you are agreeing, so your program continues. So that is what this statement means. उसको तो sidetrack कर दिया. You want to create problems? Create, not an issue. But we are welcome and we are going. So this is allow a particular excitatory input to be inhibited without affecting other excitatory synapse to have the effect on the postsynaptic cell. So if a group of 5 में one says tries to bully others की presynaptic inhibition, fine, you tolerate, you decrease it once, you decrease it twice, you decrease it thrice. What happens the fourth time? Coolly you remove that and rest 4 or 5, enjoy the party, right? So this is what it means, that it does not affect the other excitatory synapse. So the neuron causing the presynaptic inhibition must fire earlier than the excitatory. पार्टी पे जाने के बाद प्रॉब्लम नहीं करना, जाने से पहले कर लो, right? Don't spoil the mood after going to the party. So that is what it means. Causing presynaptic inhibition must fire before the entire program takes place, and it should last longer. That person should be so rigid कि नहीं ऐसा नहीं होगा, तो उसका इनहिबिशन कुछ काम आया. आपको क्लास में आने के लिए कितना मोटिवेट करना पड़ता है, हमको करना पड़ता है कि नहीं? आओ अटेंड करो, हम तुमको अटेंडेंस की धमकी देते, तुमको मार्क्स की धमकी देते, सब देते, लेकिन क्या होता है? नहीं तो नहीं. So your resistance is very much more important and lasts longer than our motivation to attend the class. So this is a presynaptic inhibition. But this will not be good in long run. What will happen? We'll give you the result, whether it is IPSP or EPSP. That is according to your function. What you wanted, you will get. Then you have prolonged modulation of the synaptic activity. Now what does that mean? I just said that एक बार आप मान लोगे, 2 बार अटेंडेंस शॉर्ट हो जाएगी. अभी मैंने जो अटेंडेंस ली उसमें 75 बच्चे गायब हैं. एक बार हम बोल देंगे, 2 बार हम प्यार से बोल देंगे, मॉड्यूलेट करते रहेंगे, तीसरे बार प्रिंसिपल ऑफिस से लेटर आ जाएगा. So again, this is how it works. So presynaptic inhibition, these are the properties, and then contribute finally to synaptic plasticity means कुछ ही कर लो, हमें नहीं आना है तो हम नहीं आएंगे, ठीक है? फिर पेरेंट्स आएंगे तब देखा जाएगा, right? So am I clear about presynaptic inhibition? Have you understood? Now coming back to the technical part, presynaptic inhibition is the inhibitory impulses given by a synapse before the major synapse, which contributes to the inhibition at the main synapse, right? So, but at times that inhibition is sidetracked and the excitatory impulses are more strong that the synapse is not having any inhibition. But if it is for a longer duration, if it is more stronger, if the modulation is more, the presynaptic inhibition works. Okay? So presynaptic, now we will talk about facilitation. We have finished about the inhibition, everybody is understood. Now we want it to be motivated, and that जो नाराज हो गया था प्री-सिनेप्टिक, we are trying to bring back. So reduce the conductance, उसके हिसाब से प्रोग्राम बना लो, तुझे जहाँ चलना है वहाँ चलते हैं. That is what you do, yes or no? So that is, you try to reduce the resistance, you try to be more friendly, and you have more sodium influx, which leads to depolarization and thus presynaptic facilitation. Clear? Yes or no? If a question comes on presynaptic inhibition, I would love to have the technical words, not these examples. But these examples were just to make you not sleep in the class, to make your imagination run कि हाँ आप दोस्तों में ऐसा ही होता है, घर पे भी ऐसे ही होता है. घर पे 4-5 पार्टी के दिन जाते हो, डिनर के दिन जाना होता है संडे को, कितनी लड़ाई होती है पहले ये बताओ. ये एक नेचुरल फिनोमेना हर घर में होता है, मेरे घर में भी होता है. So what is that? Finally, head of the family says यहाँ जा रहे हैं, फिर चाहे गुस्से में चलते हैं, चाहे बैठ के चलते हैं, या कैसे भी जाते हैं, right? So end result is excitatory, and then finally you take pictures, and then you are happy because your Instagram is flooded with likes. So that end result was the excitation, right? Okay. So have you understood all about excitation, inhibition, synapse properties? Okay. So we come to the next point. I'll just introduce this chapter. Neurotransmitters. Neuro means nervous, transmitter means a transporting mechanism. So these are the chemical mediators. अब ये बहुत ज़रूरी है, because when we learn the CNS part in depth, when we learn about basal ganglia, we learn about the various CNS disorders. We will be talking about the GABA, gamma, glutamate, glycine. So what are these? These are not merely words. We talk about serotonin commonly. We talk about dopamine. We talk about adrenergic. We talk about noradrenergic, acetylcholine, cholinergic. What are all these? These are the chemical mediators which are released at various parts of your brain time to time to have the responses as desired. Right? So chemical mediators at the synapse must be present at the presynaptic terminal. It should be synthesized by the neuron, and it should be only released on proper stimulation. If it is released continuously, there is a problem. It's not physiological. Physiological होता है कि it maintains the atmosphere, and if applied from outside, it has to have the same effect. So if we give some adrenaline, if we give some local acetylcholinergic drugs or something, it should have the same response if in situ the acetylcholine was released. So can you give me an example for this? Myasthenia gravis. क्या होता है उसमें? एंटी एसिटिलकोलिनेस्ट्रेज हम देते हैं. क्या होता है? एसिटिलकोलिन को वो डिस्ट्रॉय करते हैं. क्लियर? सो व्हाट आर वी गिविंग? देन वी गिव देम द मेडिकेशन की एसिटिलकोलिन के रिसेप्टर जो एसिटिलकोलिन को डिस्ट्रॉय कर रहे हैं उसको हम अलग रखते हैं ताकि एसिटिलकोलिन जाके कंबाइन हो एंड यू गेट अ रिस्पॉन्स. सो आई थिंक सो वी विल stop now. Classification of neurotransmitter is your next class. Small, what are the various types of neurotransmitters? क्योंकि ये chapter भले ही boring है, it's totally theoretical, but it is a very important chapter. ये आपको super speciality तक क्या ये lifelong आएगा. यहाँ पे जो भी physician, जो भी surgeons रहेंगे, जो भी इधर है, so you have, these are the few listing of the